In 2019, Australians began dumping tons of rocks straight into the sea. It looked like they were simply creating a pile of debris on the seabed. But it was an experiment. Biologists were hoping that oyster larvae would settle on it. 50 million did, in a single summer. And that turned out to be a smaller surprise than what happened next. Oysters are filters. A single adult pumps up to 200 liters of water per day, clearing it of algae, bacteria, and suspended organic matter. Where oysters grow, the water is clearer, the fish are larger, seagrass survives. Where they're gone, silt, oxygen depletion, eutrophication. Since British colonization began, Australia has lost 94% of its oyster reefs. This is the finding of a systematic study published in PLOS One in 2018, 94%. Of what once existed, 1% remains in functional condition, and a few more degraded remnants beyond that. For comparison, the oysters of Chesapeake Bay in Virginia once filtered its entire volume, nearly 70 cubic kilometers, in a week. Today, the same task takes them a year. The Sydney rock oyster, Sarcostraea glomerata, does not grow on mud. It needs a hard surface: rock, shell, another oyster. Such surfaces once existed everywhere. Reefs accumulated across generations. Live oysters grew on dead ones, on their shells, on fragments, building structures several meters tall that held for centuries. Then came the dredges, then molluscicide, then pollution, and the reefs disappeared. With them disappeared the hard surface on which new larvae might settle. The estuary became empty, not because there were no oysters, but because there was nowhere for them to attach. This is a closed loop that doesn't break itself. The standard response for decades had been one: take oysters from a hatchery, plant them by hand, protect them, repeat. This worked where larvae had disappeared from the water entirely. But it was expensive, required constant human presence, and reefs that grew this way rarely became self-sustaining. They existed as long as the budget did. Port Stephens offered something different. Remnant reefs remained there, small, degraded, but alive, which meant larvae were still in the water, simply without somewhere to go. If they were given somewhere, perhaps the rest would happen on its own. The question was whether rock on the seabed would be enough for that. Charlotte Jenkins, a biologist with the New South Wales Department of Primary Industries, had spent years studying what remained of oyster reefs in NSW. She knew the numbers. She knew the mechanics of the closed loop, and she had arrived at a conclusion that is difficult to defend on paper: that in Port Stephens there was still a chance, if larvae were given a surface, they would do the rest themselves. But before the first rock touched the bottom, she had to convince the people who had something to lose. Warimi are the traditional custodians of the land and waters of Port Stephens. They had gathered oysters here for thousands of years before the first British ships arrived. Later, their families became the foundation of the region's oyster industry. The Ridgways, Russells, and Mantons worked the farms from the early 1900s. For Warimi, oysters are not an ecosystem service. They are a connection to place that existed before colonization and survived after it. The conversations lasted not 1 quarter or 2. The goal was specific: lay 6.4 hectares of artificial reef substrate in the Mamia and Garua estuaries, the first project of this scale for the Sydney rock oyster in Australia. Not an experiment across a few square meters, as in January of that same year. A full reef designed to self-reproduce without ongoing human intervention. If it worked, it would change the approach to restoration across the country. If it didn't, $3 million and years of negotiation would become a pile of rock on the seabed. Jenkins's team came to meetings, heard the objections, returned with revised site maps and revised protocols. Fishers worried the reefs would block their traditional fishing zones. Oyster farmers worried that restoration would introduce disease or competition. Warimi wanted not merely to agree, they wanted to be authors of what was happening in their waters. A joint management protocol was signed only after each of these conditions had been reflected in the documents. Alongside the negotiations, the Shuck Don't Chuck program got underway. The logic was simple. Shell is the most natural substrate for oysters. Reefs once consisted of layers of dead shells on which live oysters settled. If shells were collected from restaurants, passed through quarantine, and returned to the water, it would be cheaper than quarrying rock, and in theory, closer to how reefs built themselves. Restaurants responded. Instead of throwing shells into the bin, they handed them into collection containers. Then came the logistics that are easy to underestimate. Each batch of shells went through a 6-month quarantine to prevent pathogen transfer into the wild water. Then a hatchery, where they were prepared for deployment. Then transport by barge. In states where oyster aquaculture existed, the chain closed. In states where it didn't, it broke at the first step. No one to receive the shells, no hatchery, no cold storage. Shuck. Don't Chuck turned out to be a program that works only where infrastructure already exists. And where it doesn't, where restoration is actually needed most, it couldn't get off the ground. This didn't kill the project, but it forced a rethink of one of its foundational assumptions. When the negotiations ended and the protocols were signed, it was time for the operation that had been planned for months. The barges entered the Mamia estuary in 2019. 3,300 tons of sandstone, not fine gravel, but large fragments offering an irregular surface of crevices and hollows where larvae could shelter from the current. The choice of material mattered. Rock that is too fine silts up. Rock that is too smooth provides no foothold. Added to the sandstone were 180 cubic meters of recycled shells from Shuck Don't Chuck. The material was dropped in layers with controlled distribution. Total area, 6.4 hectares. The operation ran for several weeks. Barges entered in rotation. The depth and current in the Mamia estuary limited the weight per entry. Divers descended after each drop to check the distribution of material on the seabed. The rock should not settle into a single flat layer that would quickly silt over. What was needed was an irregular, three-dimensional surface with voids and ridges. Where the distribution was too even, the operation was adjusted directly from the deck. Where current shifted material away from planned coordinates, the team recalculated the next drop point. None of these parameters had been specified in the original plan with this level of detail. They emerged in the process, underwater, in conditions of limited visibility and the time pressure between tides. Within a few weeks, what was meant to become a reef had settled on the bottom. Or not, and Jenkins knew this better than anyone. She had spent years convincing people it would work. Now all that remained was to wait. Oyster larvae spend 2 to 3 weeks in the water column in their planktonic stage. In that time, they drift with the currents, detect chemical signals from the seabed, and choose where to settle. Or they don't, and they die. The process is entirely beyond human control. Jenkins and her team could lay the substrate. What happened next, they could not influence. A larva that has settled never moves again. It secretes cement from its byssus gland and attaches permanently to a surface within hours of first contact. If the substrate is right, it begins building a shell and growing. If not, it dies where it landed. It has no more choice. But while it is still in the plankton, it chooses actively, responding to chemical signals from adult oysters to surface texture to light. This is exactly why the remnant reefs in Port Stevens were critical. Without their chemical signals in the water, the larvae would have had no orientation. Rock gave them a surface. Living oysters gave them a signal. Together, conditions that had not existed in the estuary for decades. Something else became clear at the same time. The fishers who had agreed to the joint protocol were now watching with interest what was happening beside their traditional zones. Some with support, some waiting to see whether what had been promised would materialize. The relationships built across years of negotiation were now being tested by whether the reefs would actually appear where they were promised. The first summer after laying, between the moment the barges departed and the moment the team could conduct the first systematic monitoring, several months passed, and throughout that time there was no way to know what was happening on the seabed. Larvae either settled or they didn't. There is no intermediate data. An oyster either attaches to substrate in the first hours after settling or it dies. The only way to check was a dive and a manual count. Until then, only indirect signs. Changes in water clarity, the behavior of fish near the sites, temperature readings from sensors. None of this was proof. Jenkins and her team waited. For her, this waiting was not abstract. She knew each of the 3 parties that had signed the protocol. She knew the fishers were skeptical. She knew the Warimi were watching. And she knew that if the larvae didn't settle, there would be no second chance to convince any of them to try again. When they finally dived on the sites at Mamia and Garua, what they saw fell outside any conservative forecast. On the sandstone substrate at Mamia, 1,790 settled larvae per square meter all arrived on their own. Across the entire site, 50 million oysters, and not one had required manual planting. But the surprise was not only in the scale. Rock turned out to be a better substrate than recycled shell, and this contradicted the logic on which part of the program had been built. Shell seemed more natural. Oysters grow on oysters, they always have. But data published in BMC Ecology and Evolution in 2024 showed that the density of juveniles on the sandstone substrate at Mamia was significantly higher. Why is still under investigation. Possibly the micro-relief of the stone offers larvae more attachment points. Possibly there are chemical differences in the surface. There is no answer yet, and that means future projects must account for something not yet understood. At the same time, and this became the sharpest management challenge of the first year, fishers began catching fish directly on the reefs. Fish had indeed arrived. The population at the sites grew by 10%, overall biodiversity by 15%. The reefs were working. But fishing gear on a young reef means mechanical damage to a structure that has not yet had time to solidify. How to protect the reef without breaking the relationships with the people who had been partners from the very beginning? No protocol had a ready answer. The team sat down to negotiate again, this time not before the project, but inside it. The Warimi held a smoking ceremony on the water. The two reefs received names, Bindayimaguba Ninang and Garuwaguba Ninang. The barges were gone. The reefs remained. The reefs began filtering water at 7.5 million liters per hour. But to understand what that means physically, you need to picture what is actually happening inside each oyster. An oyster doesn't simply pass water through itself. It actively pumps it through gills covered in microscopic cilia that beat continuously, creating a current. The gills capture suspended particles, phytoplankton, bacteria, organic detritus, and direct them toward the mouth. What doesn't suit in size or composition, the oyster expels as pseudofeces, bound clumps that settle to the bottom and become food for benthic organisms. What is useful gets digested. The rest is not wasted. This is a closed recycling loop that runs without interruption as long as the oyster is alive. One adult individual filters up to 200 liters per day. 50 million oysters, 10 billion liters every day. 3 Olympic swimming pools every hour. The first visible changes in the water Jenkins's team recorded within the first season. Clarity increased, where previously visibility was limited to a few centimeters due to suspended organic matter and microalgae. The seabed could now be seen through the water. This is not an aesthetic effect. It is a functional signal. Clear water means light reaching the bottom, and light at the bottom means one thing: seagrass can grow. Seagrass, Zostera mulleri in the case of Port Stevens, is one of the most productive marine habitats on the planet. It stabilizes bottom sediment with its roots, sequesters carbon 35 times faster than tropical rainforest on the same area, and serves as spawning ground and shelter for dozens of species of fish and invertebrates. But it only grows where there is light. In an estuary where the water had been turbid for years from suspended organic matter, seagrass had been retreating. Now the conditions had changed. The appearance of seagrass on sites adjacent to the restored reefs became one of the most unexpected results, and one of those that had not been included in any of the project's original success indicators. The reefs were planned as filters and as structure for oysters. That they would also restore conditions for seagrass growth turned out to be a consequence that no one had set as a goal. This matters for one simple reason. Seagrass and oyster reefs reinforce each other. The grass absorbs nutrients from the water, reducing the eutrophication that kills oysters. The oysters filter the water, giving the grass light. The system that humans started with rock and shell began independently restoring connections that had existed before the reefs disappeared. Fish came for both the seagrass and the reef simultaneously. The population at the sites grew by 10%, overall biodiversity by 15%. The system started with rock and shell was restoring connections faster than anyone had planned. The total cost of the project, $3 million. The social net present value by standard ecosystem services assessment methodology, $40 million. Cost-benefit ratio, 14:1. This is not abstract ecological arithmetic. It is the language in which you can speak to ministries of finance, and one that conservationists rarely have the opportunity to use. But there is a detail that doesn't enter that arithmetic. In states where oyster aquaculture is absent and there is no shell to recycle, Shuck, Don't Chuck never launched. Restaurants were willing to donate shells. The infrastructure wasn't there. This means the Port Stevens model doesn't transfer automatically to where the need is greatest, which means the 40 million NP vibe is the result of a specific place with specific conditions. What the same thing would cost where those conditions don't exist is an open question. The Reef Builder initiative today covers 13 sites across Australia. The goal is to restore 30% of the former oyster reef range. That is an ambitious figure, especially remembering that the starting point is 94% loss. But there is a question that doesn't get raised in donor presentations. Grants have end dates. Port Stevens received funding, and the reefs are alive now. But an oyster reef requires monitoring, predator management, management of fishing pressure, and in the event of damage, substrate reinforcement. Who will do this 5 years after the grant ends? Jenkins and her colleagues speak about this openly. A long-term funding model does not exist. There are options, payments for ecosystem services, carbon credits, fishing levies, but none of them is an established mechanism. This is not a project failure. It is the limit of what any project can solve on its own. The reefs at Mamia are growing, the oysters are filtering the water, the fish are returning, but the question of who will still be present when the grant runs out remains open. Today, the Reef Builder initiative is not only Port Stevens. 13 sites, several states. The methodology worked out at Mamia and Garua is now being tested in places with fundamentally different conditions, stronger currents, shallower water, and different bottom sediment types. What worked in Port Stevens does not transfer automatically. Every new site begins with its own hydrological analysis, its own community negotiations, and its own question: are there remnant reefs here? Are there larvae in the water? Is there anyone prepared to wait for results? Where the answer is no to even one of these, the methodology needs an adaptation that doesn't yet exist. 2024 brought one more detail worth recording. Data from BMC Ecology and Evolution confirmed that the best substrate for restoration is not necessarily the most obvious one. Rock beat shell, not because someone decided that, but because the oysters decided it themselves. This changes the logic of future projects and raises the question of whether it's worth revisiting the protocols in places where restoration is already underway using the old approach. Beyond Australia, the results from Port Stevens are being watched more closely than it might appear. Oyster reefs have disappeared not only in Australia. The same 2018 PLOS 1 assessment shows this is one of the most degraded marine habitat types in the world. Most restoration programs still rely on manual planting of mollusks, more expensive, slower, and less resilient than natural settlement on prepared substrate, which occurred at Mamia on its own. If this model can be replicated under other conditions, it changes not only Australian protocols, but the if remains open for now. In 2025, Australia faces a choice that confronts any country trying to restore what was destroyed over 200 years. How much it will cost, who will pay, and what will happen once attention moves on to the next project. Port Stevens showed that a reef can be restored, that 50 million oysters can settle where they are given a hard surface, that nature responds when asked the right way. But Bindayamaguba Ninang and Garuwaguba Ninang, reefs with names, cannot wait forever.